DNA is a nucleic acid and has a double helix structure. DNA is composed of monomer units called nucleotides. Nucleotides are comprised of three major parts: the pentose sugar, which is deoxyribose in DNA molecules, a phosphate group, which is bonded to the sugar at the 5' carbon, and a nitrogenous base. In DNA, nitrogenous bases can be adenine, thymine, cytosine, and guanine. Complementary bases bond together by forming hydrogen bonds, and nucleotides bond together by forming phosphodiester bonds between the 3' carbon and the phosphate group. Each strand of DNA has a 3' end and a 5' end. DNA replication takes place during interphase. In this case, the top strand is in the 3' to 5' direction, and the bottom strand is in the 5' to 3' direction. The first thing that happens is DNA helicase breaks the hydrogen bonds between the bases, and the DNA molecule essentially unzips into two strands. Then, DNA gyrase reduces the torsional strain and stabilizes the DNA molecule. Next, single-stranded binding proteins bind to the DNA strand so that they can prevent them from rebonding. DNA's leading strand, which is the top one in this example, is continuously synthesized towards the old fork, and the lagging strand, which is the bottom one, is synthesized away from the replication fork. To zoom in on this process, primase produces a short RNA primer on each template strand. Then, DNA polymerase III attaches to the 3' end of the RNA primer, and as it moves from the 3' to the 5' end, it joins nucleotides with their corresponding bases on the template strand. The lagging strand is not continuously synthesized, but copied in sections. These new sections of DNA are called Okazaki fragments. Finally, DNA polymerase I removes RNA primers and replaces them with DNA nucleotides. DNA ligase joins the Okazaki fragments on the lagging strand together by creating phosphodiester bonds between the 3' sugar and the phosphate group. The two new DNA molecules that are created during this process are semi-conservative, since each has one strand that comes from the original DNA molecule. RNA is a single-stranded nucleic acid composed of a pentose sugar, called ribose, a phosphate group, and a nitrogenous base. The nitrogenous bases in RNA can be either adenine, uracil, cytosine, or guanine. RNA also has a 5' and a 3' end, just like DNA has. In transcription, a DNA molecule is separated into two single strands by RNA polymerase, which binds to DNA at the start of a gene and pairs up RNA nucleotides with their complementary bases. Following this, covalent bonds are formed between RNA polymerase and RNA nucleotides. The antisense strand is the template for mRNA, and the sense strand has the same base sequence except thymine is substituted for uracil. Now that this is complete, the RNA separates from the DNA, and the DNA reforms into a double helix. Next, the mRNA exits the nucleus through nuclear pores and is transported to ribosomes for protein synthesis. Translation takes place at a ribosome in either the cytoplasm, mitochondria, or on the rough endoplasmic reticulum. Ribosomes are made of ribosomal RNA, or rRNA, and proteins. Ribosomes consist of a small and large subunit. mRNA is made up of triplet bases called codons. Each codon codes for a certain amino acid. The codon AUG codes for methionine, and that signals for the start of the polypeptide chain. tRNA carries specific amino acids, and each tRNA molecule has a 3-base anticodon which is complementary to the mRNA codon for that amino acid. The tRNA attaches to the mRNA and brings the amino acid methionine to the ribosome to start the polypeptide chain. Then, another tRNA molecule brings the next amino acid that corresponds to the next mRNA codon, and attaches to the mRNA at a second binding site on the ribosome. After this, both the amino acids are combined during a condensation reaction, which results in a covalent peptide bond and a dipeptide. An enzyme breaks the bond between the first amino acid and its tRNA molecule. The ribosome then slides along the mRNA so that the next codon can be read and the tRNA released so it can find another amino acid. This entire process is repeated until a stop codon is read, which can be either UGA, AUG, or UAG. Then the polypeptide will break off and be released.